Hello everyone. So in this video I will be talking about the potassium sparing diuretics and the last class of diuretics, that is the osmotic diuretics. In previous parts, starting with the first one, I have discussed about the carbonic anhydrase inhibitors, then loop diuretics, then the previous video was about the thiazides or thiazide diuretics, and this video is about the potassium sparing diuretics, and then I will talk about the osmotic diuretics, somewhat about the osmotic diuretics. So starting with the potassium sparing diuretics, which is the fourth class of the diuretics. So the potassium sparing diuretics. So in potassium sparing diuretics, as far as drugs are concerned, we have spironolactone, which is very famous one. So the spironolactone, also amiloride, triamterene, so these are some drugs. Also you can add eplerenone, so these are some drugs under the category of potassium sparing diuretics, and the spironolactone is the more famous one in these categories of potassium sparing diuretics. So let's talk about the machinery and how it is interfering with the machinery of the physiological machinery inside the cells. First of all, potassium sparing diuretics, where they are working on the last part of the nephron, that is the collecting duct, where only 1%, 1 to 2% of the sodium gets reabsorbed back into the general circulation or blood. So let's look over the one cell of the collecting duct and check out the physiological machinery and how potassium sparing diuretics are interfering with that and helps in the diuresis, and why they are called potassium sparing diuretics. So here I am drawing one cell. So this is one cell. Here, this is the basolateral side of the cell. So this is the basolateral side of the cell. Here is the blood. And this is the luminal side of the cell. This is the basolateral side, this is the luminal side of the cell. So this is the lumen. And here is the blood. Now, and this is the cell of the collecting duct, one portion of the collecting duct. Now here is the sodium channel. So here is the sodium channel, for example, through which sodium ions are getting inside the cell. So sodium ions are getting inside the cell with the help of this sodium channel. Now on the basolateral side, as you know, we have one more ATPase or cotransporter, sodium potassium ATPase, which is sending sodium outside and bringing in the potassium ions. Okay, and here is the potassium channel, which is bringing the potassium ions outside the cell into the tubular lumen. So also the chloride ions, now these are the adjacent cells, for example. These are the adjacent cells. Now what happens physiologically, that sodium ions goes inside the cells, and with the help of the sodium potassium ATPase, they get into the blood circulation or general circulation. They get into the blood and getting reabsorbed over there. In exchange with the sodium, potassium ions are coming out, and potassium ions eventually goes out into the tubular lumen from the cell with the help of potassium channels. Along with the sodium that is going transcellularly into the blood, chloride ions goes into the blood paracellularly. Paracellularly, I've already mentioned about the paracellular way of transport and the transcellular way of transport. I've discussed about that already. So sodium ions are going inside the cells transcellularly, and the chloride ions are going into the blood circulation paracellularly. So how the potassium sparing diuretics works, that they works with the two methods. First method is that they simply block the sodium channels. So if they will block the sodium channels over here, what will happen? The sodium will start appearing in the urine. Sodium starts to accumulate inside the tubular lumen. Along with that, chloride ions, as they were passing paracellularly into the blood or general circulation, they will also start accumulating inside the tubular lumen. So sodium and chloride ions, which makes salt. So salt along with urine will appear and will excrete out through the collecting duct in the form of urine. So that's how, this is the first method, that how they are working on the sodium channels and how they are helping the body to get rid of the water and initiate the diuresis process. Second method is that they are aldosterone antagonist. So aldosterone from the blood, so this is for example aldosterone from the blood goes inside the cell, and there it will interfere with the DNA. So the aldosterone will go inside the cell, they will bind to the aldosterone will bind to the receptor. This is a physiological condition, that aldosterone will bind to the receptor and will help sodium to get inside the cell and helps the sodium to get reabsorbed back into the general circulation. Now potassium sparing diuretics, what they do and why they are called as potassium sparing, because they spare the potassium. They spare the potassium inside the blood. So they won't allow potassium to get inside the tubular lumen and get waste in the form of urine. So how they are sparing? They just simply block the aldosterone receptors. So the second method is that they block the aldosterone receptors. That's why this method is called aldosterone antagonist. Aldosterone tends to retain sodium and water inside our body. So aldosterone, as they are retaining the sodium and water and increasing our blood volume, increasing fluid volume, potassium sparing diuretics, potassium sparing diuretics, what they do, they just block the aldosterone receptor and they don't allow aldosterone to bind with them. So aldosterone will not be able to get bind to the intracellular aldosterone receptors, which will lead to, so that will cause the, as aldosterone was retaining sodium and water, they will not be able to retain sodium and water in proper amount. So sodium ions will not be able to get inside the cells due to the blockage of these aldosterone receptors. Because aldosterone, as they were binding to the receptors, they were initiating this movement of sodium ions from the tubular lumen to the cell and eventually getting reabsorbed into the blood. So sodium ions will not be able to get inside the cells. Further, chloride ions will also not be able to get inside the cells due to the blocking of this aldosterone receptor intracellularly. So the sodium ions and chloride ions will appear in the urine. So that's how also they are helping in the diuresis process. Potassium sparing, these are called potassium sparing because as they are blocking these receptors, potassium ions will not be able to go outside the cells into the tubular lumen, so they will retain the potassium, due to which hyperkalemia can also be seen in such patients as this adverse effects. So potassium ions can be increased, hyperkalemia, also in females menstrual irregularities, and in males gynecomastia have been seen as they interface with the testosterone receptors too. So this is about the potassium sparing diuretics, the two mechanisms of these diuretics, potassium sparing, how they spare the potassium, how they are interfering with the aldosterone. So this was about the potassium sparing diuretics. Next one is the osmotic diuretics. So let's proceed to the osmotic diuretics, what they do. So the fifth one and the last one is the osmotic diuretics. Osmotic diuretics. Mannitol and urea are generally used as drugs. Mannitol, urea can be used as the osmotic diuretics. Now how they work, they just simply follows the osmosis rule, that from the lower concentration water tends to goes into the higher concentration. So inside the cell there is a low concentration, and outside the cells in the tubular lumen there is the high concentration. So water tends to follow that rule, and according to the osmosis, from the lower concentration which was inside the cells, and in the tubular lumen there is the high concentration. So water will move into the tubular lumen, and water will coming out into the lumen, and more water will go through the tubular lumen and eventually through the DCT portion collecting duct, and will pass through the collecting duct and will excrete out, water will excrete out in the form of urine. Now volume overload can be seen in such patients. Also the electrolyte imbalance, some kind of electrolyte imbalance can be seen. For example, sodium or potassium in higher concentration or in lower concentration. And dehydration can also be seen, as in the overdoses of osmotic diuretics in mannitol or with the urea, more water, excess amount of water can be appear in the tubular lumen from the cell, and they can appear in urine. So that's why dehydration is also the adverse effect seen with such drugs. So they can be given with the loop diuretics in the combination actually. So this was about the osmotic diuretics. This just simply follows the rule of the osmosis. So with this video, this is the conclusion of the diuretics. I have talked about all the classes. First one I talked about the carbonic anhydrase inhibitors, then loop diuretics. Third was the thiazide diuretics. Fourth one was the potassium sparing diuretics, and the last one is the osmotic diuretics. And this was all about the diuretics. I hope you like this stuff. Thanks for watching.